I took all the way today. I enjoyed it. I didn't do terrible. I mean, like, all of the. I felt like we watched more class. Like my lab, and then to the end, we remember time on it. Can I argue that? Or
Right.
because you can say what the OLED is done. But you can't say R. You can say R here or R. Because everything is, but I don't know. Yeah, exactly.
Okay, cool. So we are ready to go for today. So last time when we were talking, we had finished up by talking a little bit about, we talked through a bunch of our different Cambrian fauna, looked at some examples. I'm clicking through essentially the last slide here. Okay, we looked at the tree of some of these fossils and how they map onto our current tree. Talked about the Burgess Shale, how it formed. We're talking a bit about its environment, and then tacked on thinking a little bit about ocean environments, because for a while we're going to stay in the oceans and be thinking about adaptations to these ocean ecosystems. So our final little bit, last slide that I'm leaving in this lecture, is about that preservation piece. So when we're looking at that preservation, we know that this shale is forming with mud, sediment layers coming off the land, settling onto the ocean floor. We know that we find most of our fossil deposits here at the bottom of this cliff. So maybe it's a little bit protected by this cliff. It's also down at the base here. So maybe we have more sediment watching. But other things that we know here actually that are probably improving this fossilization process as well is that down here at the base of this cliff, we see oxygen depletion. And that's relevant to our fossilization process, partially because of why fossils don't form, right? So if you think about in modern life, right, some animal dies by the side of the road or in the woods, right, it's probably not going to become a fossil. Part of that is because it decays, right? And so that decay has to happen by other organisms eating and processing the remains of that organism. So this oxygen depletion at the bottom of the ocean here means that a lot of those microorganisms that would usually be part of that decay process can't survive and aren't there. And so this means that the things that settle on the bottom there kind of are more likely to stay complete and still be there to get fossilized. Actually, heard, I haven't fact checked that this is true. I heard that it's cold enough at the bottom of Lake Superior that this happens to a certain extent as well, that decay processes are slowed down or not present. I heard this from someone who's very freaked out that with global warming, maybe dead bodies will start rising to the surface of Lake Superior. I don't know if that's a real concern, but kind of a similarly based concern to what we're seeing here when we think about why these fossils are forming. The other thing that's true is that sediment here at the bottom of the cliff is full of clay. And there are some people who think that this clay may have inhibited the bacterial growth and therefore inhibited that decay process. The other thing is that we have exoskeletons on some of these fossils, and the fossils themselves have these thin carbon films that got partially replaced by clay and iron-rich minerals. So we have this permineralization process going on there. Some of them, we actually have their gut systems partially preserved, which is kind of cool. And this is because of basically high amounts of phosphate in some of those digestive systems. So they go through a different sort of fossilization process with that phosphate called phosphatization. Okay. So that's how these fossils are forming, right? So now we're going to click over. Great. And we're gonna think about part 2. Oh, I did leave that in there. Hold on. Okay. All right. So we see an awful lot of different animals appearing in the fossil record during the Cambrian, to the point that this is known as the Cambrian explosion. Anybody have any ideas why that might be? So what did we have happening at the end of the Ediacaran last time? At the end of our last unit, what did we say about all those organisms? What happened to them? Are they still around today? No, they died off, right? So one of the things that's happening as we start the Cambrian is that we saw all these shift and potentially to the point of a mass extinction in the Ediacaran. So right at that border between the Ediacaran and the Cambrian, we have something that happened that caused those Ediacaran organisms to kind of disappear. There are different theories on whether that's because they were no longer alive or because they weren't fossilizing as well. But if we go with this sort of mass extinction or even mass replacement model, this means that all of a sudden we're going to have a lot of niches ecologically available for these Cambrian organisms, which also happen to fossilize very well, in part because they're developing things like shells and exoskeleton. Yeah. So an ecological niche is basically just like a little sort of carved out position in the ecosystem where you have an animal, something that is able to eat certain types of food, catch certain types of prey, live in certain types of places. So like if you have no animals out in your environment living in caves, but you have a ton of caves that are nice and protected from the rain and the elements, right? You might expect to see something adapt to learn to use that resource, utilize those cave systems to live in. And that's something that we see throughout history, geologically, and throughout a lot of different species. We see them adapting to fill sort of blank places, right? So if you have, right, like nothing that eats mosquitoes, but there are tons and tons of mosquitoes, you might expect something to try and figure out how to eat mosquitoes because they're all over the place. This is one of the problems with invasive species, right? When we see an invasive species suddenly invading our environment, as no predators, there's nothing in that niche competing with that organism. That's part of why they can take over so well, because they're out of their original environment. Nothing's evolved to keep them in check, and so they kind of explode. So this is one theory of what might have kind of been happening when we see this Cambrian explosion or Cambrian radiation, where we suddenly see this new diversity of organisms, diversity of metazoans. So if we think that something like a mass extinction happened at the end of the Ediacaran, we suddenly have all sorts of places in the ecosystem that are no longer occupied by Ediacaran, allowing the metazoans that did exist in the Cambrian to kind of take over. We'll see that when we get to dinosaurs and mammals, right? This is part of why we have so many mammals alive now, including us, right? There were tiny, tiny mammals, a few of them alive at the same time as the dinosaurs. All the dinosaurs go extinct, except for the birds, pretty much. And then mammals expand to fill a lot of those niches. So this is one possibility. We'll think about some alternative possibilities as well involving development of a certain number of like key genes that would have similarly opened up new sort of ecological spaces in the ecosystem, new types of life that would have been available to these organisms. Okay. So here's a term we want to know when we think about these kinds of explosions or sudden spreads of new and varied types of organisms, the sudden appearance of diversity. So we often think about this concept of adaptive radiation. So adaptive, referring to new adaptation. So you're adjusting essentially and changing to fill new places in the ecosystem. So new environmental sort of niches. So formally, if we want to define it, adaptive radiation is a rapid diversification, so lots of new things, from an ancestral species, giving us many new forms. So this is usually going to be due to a change in environment or the opening of a new environmental niche. So the classic example of this, which we'll go through just to get down the basics of this topic and make sure we're all on the same page with this. Classic example is the Galapagos finches. So here we see sort of phylogeny of our Galapagos finches. So we see evolution from some sort of ancestral seed-eating ground finch into many diverse finches that we see in the Galapagos Islands. We can see that they have diverse niches. So some of them eat seeds, some of them eat insects, a couple of them eat the buds and fruits of plants, and some of them specifically eat cactus seeds and cactus parts. So this is a classic example, partially because Charles Darwin wrote about it when he was writing down the history of his voyage on the HMS Beagle. So the HMS Beagle was a ship that went to South America to chart the coast, basically, to start to build maps of South America. And Darwin got a position as a naturalist on the boat. So his job was to go and collect things and describe what was there while they were mapping out the coast. Right. So he wrote down all his notes, and this is where we get the book, The Origin of Species, from. This is where he was developing his ideas on how we end up with this diversity of organisms through natural selection. So when we think about these finches, I like this picture because this both looks like a phylogeny and like a tree for our finches to sit on. I want to think about the Galapagos Islands themselves. So these are a series of islands off the coast of Ecuador. So we can see them here. And we have a distribution of different finches across these islands. So this is from a paper, a recent paper looking at some specific gene, I don't remember, in these Galapagos finches. But we can see nicely, therefore, the finch species listed out for each of these islands. So we can see that we have a different finch here on this island, Fernandina, versus on this island, Isabella, and so on. So Darwin noticed, and I thought this quote was worth pulling, right, that he saw nearly perfect gradation of structure in this one group, traced in the form of the beak, from one that has really large dimensions, essentially, to another that basically doesn't differ from a warbler, another species of bird. So we have different species on each island, and we can see all sorts of intermediate forms. We can see a really wide variety of beaks spread across the archipelago. So this also makes a good example for another important term related to evolution, which is allopatric speciation. So in allopatric speciation, this is talking about the spatial reason for the speciation. So when we're thinking about adaptive radiation, we're thinking about movement into niches. So eating different types of food is very commonly the new type of niche we're thinking about. In allopatric speciation, we're thinking about the fact that we're starting with an ancestor that then gets separated into geographically isolated populations, right? So this has to do with the fact that we would have had some original finch making it from the coast of South America out to this island, these set of islands. But then we end up with that population getting concentrated on different islands and not really having any gene transfer or mating occurring between the islands. So this is part of what allows the new species to develop from that ancestral population. Part of what allows us to spread out is that we don't have much mating going on between the neighboring islands. If we did, we would expect sort of a gentle gradation kind of happening among the islands, but there would be enough sort of gene mixing that we wouldn't expect this to produce different species. So in allopatric speciation, the reason that we have limited mixing of the gene pool, breaking up of that gene pool into different little chunks, is because of this geographic isolation. So looking at the adaptations of these finches, the important thing to focus on for them is their dietary specialization. So like this figure I found in a paper that was comparing these finch beaks, which is really the trait people look at, although you can see that there's also variation in, for example, the eyes and some other structures. We're comparing those beaks to different types of pliers, right? So when we're thinking about this dietary specialization, to eat something, right? You eat it with your mouth, you need to bite it, right? So looking at these different beak forms, we can see that different beaks are better, basically, for eating different types of foods. So if we think about how you would eat an insect or like get nectar from a flower even, right? You might think, as we have down here, right, like a long sort of pointed beak, this pointiness, right, is going to give you some precision for poking and probing into places where you would find insects, basically. Mostly these are insectivorous stuff. Versus if we have a convex curved beak, what we're going to end up doing is putting a lot of pressure right at the tip. So that curve is basically curving our forces and focusing them downward right on the point. And this is going to create forces that are really good at like pinching off little bits of something, right? So think about biting something with your two front teeth, right? Like little nips at the edge of something. So when we think about tip biting, we're often thinking about like plucking little buds off of plants, plucking new greenery in this sort of precise manner. A lot of these finches eat seeds though, and you think about like chewing something, right? You don't chew like a rabbit, like with your two front teeth, right? That's not really effective, and you probably break your teeth, right? I have a broken front tooth, so I think about that somewhat often. So really when you chew, you move your mouth, right? You can feel that you're chewing at the back of your mouth, you're chewing with your molars, right? And this is going to be true for our birds and specifically our finch beaks as well. So if we have a beak with a deep base, so like a really wide point where it meets the skull, this is going to be better for like getting a big seed and crunching it and putting a lot of force back there in the base of the beak. And this helps you crack open hard food. So this is going to be good for eating like really hard seeds, some more difficult to eat grains. Okay. So I take a second to predict what you think each of these finches might eat, because these are great examples of some of these Galapagos finches. And you can chit chat with your neighbors when you've made your predictions. So there's one thing to hear that they're different, and another thing to try and look at a bird and see that's different.
That's the one where I'm
How are you feeling about these birds? Which one do we want to make a guess at first? So this one primarily eats insects and also does a little bit of nectar eating from the center of flowers. Cool. So that's that long, quote unquote long, this bird is small, right? But a pointed beak there. What about the other two? Both of them?
I think the one on the left.
This one? So this one is actually one of our seed eaters. Really, it eats like kind of grain. So it's possibly kind of a little bit of an in-between. So you can,
It kind of looks like it.
yeah, it totally does, right? I tried hard to find one that had a real tip biting kind of point. So what you can see here is this curve here. So this is what we mean, that curve leading to the forces of the tip. This one happens to have the tip poking out. So this is one of our vegetarian sort of bud eating finches here. But also looking at the real examples, you can see how this took a lot of detailed staring at birds, probably, right? Yeah. So when we're trying to figure these things out, it does require a lot more data collection than you might think, and a lot of meticulous data collection and cataloging of all of these birds, but also other organisms, in order to come up with these hypotheses and then test them. There are many more pieces than that, but we see a lot of gradations between them. And because we do expect them to have a common ancestor, we expect them to split off, right, into sister groups, right? We might also expect to see some evidence of that common ancestry too, right? Like, so when we're looking at those beaks, we might expect that part of that is adaptation to the new niche, but part of those traits might also be based on what the ancestor of that species is doing. So here we are looking at a phylogeny of these finches. So starting out with our common ancestor from the mainland of South America, we see separation into 3 genera, so that's plural of genuses. So we have ground finches, tree finches, and then warbler finches. So you can see these warbler finches. I got some nice pointy beaks here. And then it's the ground finches and the tree finches that we can see some sort of complex relationships related to their adaptations when we're looking at these species. So we can see these seed eaters over here, and that seed eater, I agree, does look a lot like that third picture, right? But so just this bud eater a bit over here, so we can see some relationships there. We see cactus flowers eaters, just some of the various finches, basically. Cool. All right. So at the end of the Ediacaran, we assume some type of dying off of these Ediacaran biota. Exactly how that happened, we have different models. We could have had a catastrophe and a complete mass extinction, right? Possibly due to disturbance in the global carbon cycle that we could see when we were looking at the rock record. We also thought about biotic replacement. So we saw that we had some metazoans that were present in the Ediacaran. And when we look at existing animal phyla and try and use molecular data to date when they split from each other, we do get dates that are farther back from the Cambrian. So we do get dates back somewhere into the Ediacaran. So that's a secondary form of evidence for the fact that we have some of these metazoans possibly starting out kind of soft and squishy and poorly fossilized sometime in the Ediacaran, and then becoming more prevalent and better fossilized in the Cambrian, which might be part of why we see them so diverse at that point and so kind of separate and sort of fully formed once we get to the Cambrian. So some of that biotic replacement model would have been a changing environment caused by those early metazoans, so caused by our evolution of animals, essentially. So mixing up sediment and aerating that subsurface, so aerating essentially kind of the sand at the bottom of the ocean, right? So that the benthic environment might have changed, disturbing some of the ways that those Ediacarans were living. Pumping water for filter feeding, ventilating the water column, and removing some of those ways that the Ediacaran were getting the nutrients might also have helped kill them off. And then also active predation, essentially, of those metazoans on the Ediacaran. So the Ediacaran, a lot of them we saw were kind of anchored, right? And you know that a lot of our early metazoans now, right, can move, right? So this might open up a new niche in that way, right? The ability to eat Ediacarans might have been part of that new niche as a predation. We also talked about the fact that changes in taxonomy, so the way that these fossilize, might also have been part of why we saw them disappear. But in any case, they definitely died off at some point because we don't have them anymore. So, and so here is just a picture of that carbon change, that carbon cycle change. So now we can look, instead of at the Ediacarans, at the metazoans here on this chart. All right, so as we were going into the Cambrian is the C here. So at the end of the Ediacaran, we had a lot of different Ediacaran biota, a lot of diversity there that starts to narrow off and then disappear when we get to the Cambrian. This is seems to be linked with some shifts in our delta 13 carbon ratio here. So this might be some sort of change in the environment messing with the Ediacaran. The other thing that we see is we see this explosion of the diversity of metazoans as we move through the end of the Ediacaran. We can see that we have some of these lineages appearing, diversifying a little bit, and then in the Cambrian, they're really exploding. So now we know specifically a fair bit more about this example, the Bilateria, right? So those were our bilaterans, our organisms with bilateral symmetry, so including us, right? We can see that they really are taking over, starting at the beginning of the Cambrian, also moving through. So these are some of our other metazoans. The Porifera, the sponges are, you know, never really in charge of the world, right? So they start deeper in time. They do diversify a couple of times, including in the Cambrian, but not really taking over. But if you think about a sponge, right, a sponge is not going to like predate on much, right? Kind of stuff there. Cnidarians are like jellyfish right here as well. We start to see those expand. So it's what's kind of going on in here in our geological record as relates to the diversity of these organisms. Okay. So one thing that we do see that in the Cambrian that sort of adds for the evidence that the predation model might be part of what's going on. Part of what's leading to the diversity of organisms is we do see literally burrowing animals arising. So here we have the organism Otoya. So this is in phylum Priapulida. So it's a carnivorous worm. Yeah, so we have two things happening here, right? So we actually find it in burrows. And if you think about why you might make a burrow, like if you're just chilling, eating biofilm and stuff that's settling on the sediment, right? There's not really a point in burying yourself under the sand. But if you start to think about a situation where you have lots of organisms moving around in these oceanic environments, you can start to see why burrowing might be helpful, right? If you make a burrow, you're probably doing one of two things or maybe both of them, right? So to make a burrow, you might be hiding from something, right? Hide yourself under the sand. Given this is a carnivorous worm, right? You might also be hiding under the sand, not to escape predators, but to hide so your prey don't see you, so you can then burst out and eat them. So we think that may be some of what this carnivorous worm was doing here. So these burrows. We can also see evidence of predation from like trackways. So we have trace fossils. They're not actual organisms, but like tracks that they literally made in the sand, and then those tracks got fossilized. So some of them we see like two things walking along in the sand, they meet, and then only one walks away, right? Which is pretty clear evidence that something happened there in that moment. Yeah. Okay, which leads to another evolution biology term that I would like us to know and be able to think about. So there's this idea called the Red Queen hypothesis. So this is an explanation for diversity we see. This is often talked about when you think about like immunology and parasites and microbes and things like that. But when we're thinking about the evolution of predation, I think it's pretty relevant here. So the idea here is that if a species just stays the same and never adapts, something is going to adapt to outcompete it or to eat it, and then it will end up extinct, right? Because it's not going to survive. So some of the explanation for evolution here and changes that we see in the fossil record and in modern life is that we have this need to constantly adapt and evolve in order to avoid getting outcompeted and to avoid getting completely predated upon. So we have this need for constant change. So organisms responding to their stressors, and then the stressor, right, is another organism, it responds, and we have this whole back and forth. So the name of the term comes from Alice in Wonderland. So the Red Queen always running in place, in the same place, and always commanding them to do different things, right? So this is the Red Queen hypothesis. So here's a model of like what that would look like with just like a rabbit and a fox, right? So foxes eat rabbits. So as the rabbit evolves more speed to get away quicker from a fox, you would expect fox populations to kind of drop because they're getting hungry, right? They can't get enough food. If they don't evolve or adapt in some way to that change in their prey organism, right, we'd expect that decline to continue. But instead, what we see a lot more often is that we then have an adaptation to basically beat that defensive improvement. So in this case, the fox gets faster itself, so now it can catch the rabbit again. So we have this sort of cycle back and forth between predator and prey, creating changes overall in each organism. Okay. So a useful fossil for us in the Cambrian that allows us both to look at diversity as well as to kind of track changes in time are trilobites. So these are, so they're commonly considered an index fossil. So an index fossil is one that's used to define geological periods. So we can date rocks with things like zircon, right? But another way to get dates of rocks is to look at the fossils you're finding in that layer, right? If you've dated it somewhere else, you know when those fossils appear and disappear in time, right? You don't need to go through the expense, the effort of necessarily testing every single sample if it's good enough for you to just kind of know generally what time period you're in. At least it gives you a good start, because then you know what type of testing you might need to do, because different types of testing are good for different time scales. So index fossils are ones that we would use to kind of date sediment layers. And the trilobites are great because they are common and widely distributed in the Cambrian. So they appear during the early Cambrian, kind of weird like the other Cambrian things, but they really do appear kind of fully formed. So there are discussions about where they came from. But when we're looking at the Ediacarans, right, we saw some things that maybe looked a little bit like a trilobite. So one idea about their evolution is that the reason we suddenly see them in the early Cambrian is that that's when they got their exoskeleton and they become an arthropod. They can fossilize a lot better. And they go extinct in the Permian. So they're around for a really, really long time. They're really successful as a clade, so as a group. And they look sort of the same for a really long time. But if we look carefully at them, we see that we have them filling all sorts of ecological niches as well. So they fossilize really well because of their exoskeletons. Arthropods. Different trilobites are clearly documented in more specific time periods as well. So you can look, if you are a trilobite scientist, at very specific features on the trilobite you have in your sediment layer, in your exposure, and that could also tell you more about more specifically what time period you're looking at. Okay, so on those pictures, I had a whole packet for you. They didn't print super well, so I just printed this big picture for us. The other activities we'll do on the slides. But I want you to take a look at some of these different trilobites and try and figure out what the important features you think you can see if you were trying to go identify a trilobite. What are some sort of commonalities you would look for? That can pass around. Who's trilobite?
General features. Oh, okay.
If you decide you love trilobites, I was just looking up how much they cost. They are quite cheap. You can very easily buy yourself a trilobite on like Etsy or something if you want.
Really?
Yeah.
How much is that, right?
The first link I clicked was $15, but it looks like if you want one rolled up, that's $4. So,
How about one like this?
oh, yeah. So when I say they're common, they're common. Okay, how are we doing on features? What are some things we can see? Yeah, they do have eyes. They do have bilateral. Yeah. Yeah, so it's pointing to this middle part. This is actually kind of how they get their name. So when I say they're trilobites, they have 3 lobes actually. So they have this anatomy where they do have that central bilateral line. I did not put that in the middle. But they have the central lobe, and then they have 2 pleural lobes, one on either side as well. You'd also want to note like their segmentation. I'm going to pull up a big picture of our trilobites. Another cool thing about them is that this one's probably an expensive one, I would guess. Some of them fossilize with minerals like pyrite, which is fool's gold. So you can literally find like golden trilobites in certain places, which is kind of neat. Okay, so we're looking at trilobites, right? So we can see, actually, let's talk about the segments first. So they are arthropods. So we'd want to be able to see that segmentation, and someone asked me about those legs, right? I don't know if you can see them on that diagram that you have, but you would expect to see those jointed legs right below, right? And those segments that we see have specific names. So we separate our trilobites into the cephalon. So ceph always means like something to do with head. So the cephalon is going to be that portion where we see this shield-shaped front of the organism. We see an eye. We actually see a hypostome. So basically it's feeding parts here. And then we see the thorax. So we got our thorax for our trilobite. When we're looking at that thorax, you do notice those 3 lobes, right? So we have this central axis, right? And then we have pleural lobes on either side. So the pleural lobes, pleura is kind of like lungs. This is where you're going to have the gills of the trilobite. And then we have this pygmalium at the back. So this is the tail structure essentially. Oh, and that central lobe is called the axial lobe. Axial is like a term of position here. So we can see this from a couple of different orientations. Usually, I don't exactly know why. We usually get dorsal views of the trilobites, but
Do they have an exoskeleton?
yeah, they totally do. They have an exoskeleton, and that's part of why they're fossilizing so well. And they actually also molt. So part of their development, like how they grow, it's like, yeah, like a cicada, exactly. Or like a snake, right? Apparently some of them like actually break out of the front of their head, like their head splits open and they crawl out the front. So they create this like, huh? It would be cool to watch. Yeah, so we also get sometimes it's like a lot of them are just going to be that fossilized exoskeleton left behind after the mold.
Does that help with fossilizing? Is it like we're able to fill with sediment and then?
You know, I hadn't thought about that, but probably, right? And that would probably also like, if you don't have guts in there, you probably decay less, right?
Yeah.
Just like we have our nice horseshoe crab here, which is not a trilobite, but definitely looks like a trilobite, right? Yeah. And this one's just dry and desiccated, right? Yeah. Really fun. Okay. So when we are looking at our trilobites, since there are so many of them and they look quote unquote so different, they're all going to have those features, but they have them in different sizes and different shapes and different arrangements. We can use some of those features to try and interpret the lifestyle or behavior that ecological niche each of them was occupying. So the trilobite, bless you, did so many different things. So even though they look similar, once you have a trained eye, you can see they're actually quite different. Kind of same situation is going on with our finches, right? Like if you don't look at birds much, it just looks like a bird. But if you spend years and years looking at birds, my mom now, every time we're on a walk and she points and I think it's something exciting and then it is a bird. But they get exciting as you know more about things, right? Like once you know more about something, you can kind of distinguish what's special and what's not. So here we have some of our inferences for our lifestyle and behavior based on some of what we see in the fossil evidence. So this is like kind of how we do this. Okay. So let's just talk about the lifestyles first. Actually, just want to make sure really that everybody remembers what the word pelagic means from last time. Remember. So that's part of why we were looking at the parts of the ocean. Yeah, so open ocean, right? So when we see the word benthic, we're thinking about like living on the sand on the bottom of the ocean or somewhere along the shelf. When we see this word pelagic, we're thinking about something actively living in the water. So that's why it says pelagic lifestyle swimming, right? So it had trilobites that just like swam around in the water, not like living on the ground. That was where they lived in the water, kind of like a fish. We have trilobites that burrow, trilobites living in quite deep water, trilobites that eat particles, trilobites that filter feed, as well as some trilobites that eat soft-bodied worms, so are predating upon some other organisms. So I want you to take a second and read through some of these pieces of fossil evidence and interpretation and try and figure out like how some of those things relate to each other. So how did we get to this inferred lifestyle or behavior from that fossil evidence? And you might want to look back at your diagram while you're thinking about this. We're now using some of those specific trilobite anatomy parts. Yeah. Okay. Oh,
It's little like deep water living, the fact that they don't really have eyes, that would make sense.
mhm.
And I can just like,
Yeah.
like can't penetrate and like swimming, shortening their body or like not
Mhm.
makes it faster and better.
It works. Okay,
So like it's cool. It kind of makes sense.
cool. Does anybody have questions about this part? Okay, great, because next we're going to try and match some trilobites up. All right, so looking at these, let's decide. Okay, 6 trilobites, and you can use that chart on the previous slide. Let's try to find some of these lifestyles in our How are we doing on these? Bigger than the table, right?
Yeah.
Yeah. Which one? A trace fossil.
But I thought.
Better not. Actually, that. Going down.
Okay, so let's do some comparison. All right, so let's look for a pelagic trilobite. So what are we looking for when we're looking for the pelagic ones? Yeah. Mhm. Who's got a guess at which one of these that would be? Okay, so what if I tell you, so when we say reduced thorax, we're thinking about the fact that it's like swimming through water, right? So think about something that's like streamlined, right? Like when you're diving, right? How do you, right? Like, so the answer is actually B, right? So this reduce that we're looking at here is actually this like narrowing, right? And you can see in profile also like narrower. So that's that like streamlined bit. Huh? Yeah, that's why we're looking at it, right? And it would be helpful if like we had some of the other things, but like if you look at these spines, right, at the front, so looking at that view, right, if you imagine like trying to walk along on the sand with a big spine like that, how would you do it, right? Because what we're looking at is a shape like this, right? So then your spines would be touching the sand and your whole body would just be doing what? Planking. So that's part of why they think it was swimming, is because they can't figure out like how that could possibly lie flat, right? So it must be not lying flat. Is kind of how we're looking at that. All right, what about burrowing? So the answer is C. And like, don't feel bad that this is hard, right? Like I looked up the answers, right? But like we're practicing how to look at them, right? And kind of relate like what people are doing when they make these interpretations. Okay, so knowing that the answer is C, when you picked A or C, what were you looking at there? Yeah, so that axial lobe here, right? So that's that center one. Yeah, it's broad and it's super smooth, right? Because if you're trying to burrow down under the sand, right, it's easier if you don't have a bunch of friction pushing against you. Right? So that's a different kind of idea of being streamlined, right? So instead of, excuse me, like narrowing for streamlining, we're thinking reducing friction, but still having a lot of strength to push away the sand, right? Which is why we have to stay kind of broad here. All right. How about deep water? What are we thinking for deep water? What grades are we looking for? Yeah, we can't really see the eyes, which would be one of the great things to look at, right? The answer is E here. Okay, so what we would be looking for are eyes reduced or absent, the wide bodies, right? So relative to their length, they're kind of wide, right? Because we got two of them here. And we have these long spines, right? So those long spines we can see here. Long spine going all the way back. Those are what those genial spines are. Oops, logic. C, burrowing, C. Now we got deep water for E. Now we have our feeding mechanisms. So cross out the ones we've already done, so I can just look. Okay, so the remaining three we have to figure out how they are feeding. Okay. Anybody feel good about any of these three, 4, 5, or 6? F for which of them?
Let's do for the grade.
I got something that thing got like spikes in the front.
Yeah, the mouth part, right? Big old mouth part. Yeah, so big old mouth part for F for eating a nice squishy worm. Right, and then we got particle feeding versus filter feeding. So for particle feeding, we're trying to like scoop the sand and pull like particles out of it. For filter feeding, we're like pumping water through. So this is the one that's a trace fossil. So what we're looking at is that scooping of the sand, right? So we're like kind of looking at the tracks kind of along. I think that's the track. Anyway, so also this species, genus was specifically named in our table as one of our particle feeders. So that is C, which leaves filter feeding for A. So what are our traits for filter feeding? Go back here too. Yeah. So this is about pits, which we may or may not be able to identify pits, but we could, if we were looking closer, see some of these extra pits along this fringe here for A. Okay. So the stuff, top line general, sometimes seems easy when you're actually trying to do it yourself. A little tougher. We'll pick up here next time and we'll talk about eyes and Hox genes next week.